Thanks very much, and thanks for everybody who stuck around to one of the last sessions of the day. I know it's been a bit of a slog, but I appreciate you guys being here. So I'm a PhD candidate at the University of Toronto, and I'm working on an evolutionary question in plants, and I'll go over some of the details of that here. And as sort of a tangent to my PhD project, I've been trying to come up with a nice neural net-based method for tidying up what's called a de novo transcriptome assembly. And how many people have some kind of a biology background in here? 2, 3. Okay. Okay, sorry. Okay, great. So you guys know what a transcriptome is. For everybody else, I've got a little bit of a primer on that, as well as some background on deep learning. So I apologize if the laptop is a little bit sluggish here. I'm using an older machine that is not mine because I'm currently between laptops. So just a brief outline of the talk, starting with a note that this is very much a work in progress. I had hoped to be able to present some flashy, look how great it is compared to some of the methods that people use in the scientific literature. I'm not quite there yet. But that means I can talk about some of the problems and pitfalls that I've had along the way, which might be more interesting for a developer crowd. So I'll talk a little bit about myself, then I'll talk about what the difference is between working with model organisms and everything else. And the reason why this matters is because with model organisms, we never have to do a de novo transcriptome assembly, which I know I haven't defined yet, because we have reference genomes. Their genomes have been sequenced. For everybody else, we don't have that luxury, and I'll explain what that means. I'll give a very quick primer on genomics, and a little bit of background on deep learning. And then I'll discuss my problem domain for this project, and close with some results thus far, and also some of the pitfalls that I've encountered. Okay, so first of all, a little bit about myself. I'm mostly self-taught with programming, like a lot of people that I've chatted with here. My first programming language was actually Common Lisp, which sort of primed me for Python in a few ways. It's a language that uses a REPL. It emphasizes rapid prototyping and later optimization. So Python was a bit of a natural fit coming from Lisp with, I don't want to say a nicer syntax because I like the weird Lisp syntax. The main thing I like about Python is the community and the availability of big packages to do almost anything that you could possibly want to do. I'm currently, as I mentioned, a PhD candidate at the University of Toronto in the Department of Ecology and Evolutionary Biology. I'm studying the evolution of something called C4 photosynthesis, which is a variant type of photosynthesis that a small number relative to the total number of plants actually performs. And it's evolved independently a number of times in unrelated plant lineages and in response to declining atmospheric CO2 over the last 35 million years. So even though humanity is rapidly pumping lots and lots of CO2 into the atmosphere, in geological time we live in a period of relatively low CO2, which causes all sorts of problems for plants, and C4 is one of the evolutionary mechanisms for getting around some of these limitations. And I'm not going to go too much into the details of what C4 photosynthesis is, but I'll talk about why it's cool and why I'm interested in it. And I use Python almost every day now for various sorts of bioinformatics tasks, whether it's just parsing data to get it from one program's output to another's input, or more complicated tasks like trying to build neural networks to solve problems. So what's so cool about C4 photosynthesis? Well, first of all, C4 plants kick ass. They have really high photosynthetic rates. They're very productive in warm conditions. Some of humanity's most widely grown crops are C4, so maize, corn, sorghum, sugarcane, and certain species of millet. So a lot of really important cereal and grain crops. C4 represents a potential route for the improvement of non-C4 crops. And so, for instance, there's tremendous international interest and a lot of money spent on trying to produce a C4 rice. And a lot of this funding comes from the Bill and Melinda Gates Foundation, because if we could produce a C4 rice, we could boost productivity, and rice being such a staple and important crop for so much of the developing world in particular, this would be a big deal for human food security. And it's also ecologically and evolutionarily fascinating. So C4 plants define huge portions of the global ecosystem, C4 grasslands in warmer climates. And C4 photosynthesis has somehow evolved independently over 60 times in unrelated lineages of plants. So it's one of the most strikingly convergent traits in evolution. And you'll see that as soon as the next slide loads. There we go. So this big hairball is an evolutionary tree of all of the flowering plants with the C4 lineages highlighted in red. And so you can see there's a few hotspots with lots and lots of origins of C4 in these groups. These are grasses and sedges for anybody who can't read that there. And then there's a scattering of other transitions to C4 elsewhere in the tree. And so my study system is right over here, a genus called Blepharis that grows in Africa, as well as through the Middle East. And the model plant, the big workhorse for C4, or for plant biology in general, is all the way over here on the other side of the tree. It's not Cleome. Cleome is the closest C4 lineage to the model plant. And the model plant is a little mustard weed called Arabidopsis. And I'll talk in a moment about what it means to be a model plant. But suffice to say, my study system is separated by about 200 to 250 million years of evolution from the system that a lot of the work on plant biology has been done in. So what do I mean? I keep throwing this term model organism around. What does that mean? Well, here are some examples to start off with of model organisms. So budding yeast, Saccharomyces cerevisiae, is a model organism for doing simple single-celled organism genetics, and actually an even simpler system is bacteria like E. coli. Fruit flies, everybody's probably heard of fruit flies being an important system for doing genetics. And in plant biology, the most important model organism is Theocress. It's a little mustard weed, as I mentioned. Scientific name is Arabidopsis thaliana. That's what it looks like. And so, what are important traits of a model organism? Well, I'll talk about the model plant because I'm a plant biologist. So they're small in size, they're easy to grow, they have a short generation time, they're self-fertile, you get lots and lots of seeds out of them without having to do anything. They have a small genome for plants at 135 million base pairs, and for comparison to ourselves, we are at 3 billion base pairs, so this is a relatively small genome. The first plant genome sequenced was Arabidopsis in 2001. You can order up on the internet mutant lines for any gene you might be interested in, so this is a fantastic resource. And there's lots and lots and lots of genomic and bioinformatic resources. So for instance, this is an ad, a little splash that appears on the homepage for the Arabidopsis information website. So you can get curated gene summaries, gene names, publications, annotations, all sorts of things, delivered fresh every week, it says. And you also have all sorts of nice tools available to you when you work with model organisms. So this is the integrated genome browser. What we're looking at here is a view of the entirety of one chromosome, and all these little black blobs all over it are genes, coding regions of that chromosome. We can zoom in and we can see information on specific genes. So you can really, in a very literal way, you can read the genome of an organism for which you have these resources available. So I'm zoomed in on a gene here and I've moused over this one and we're seeing, okay, it's a GPI transaminase subunit, whatever that means. There's also all sorts of other tools available online. So this is one where you can see the expression of any gene. You just throw in the gene name and you get a nice heat map based on the plant. You can see what tissues it's most expressed in. So this gene that we're looking at, for instance, is most expressed in mature seeds. So given that all of these resources exist for model organisms, why the heck would anybody want to work with a non-model organism? Well, for what I'm interested in, it makes sense because C4 didn't evolve in Arabidopsis, so how could we possibly study C4 evolution in this model plant? There are C4 species with genomic resources. So for instance, the genome of corn has been sequenced. And also a number of the other important cereal crops are sequenced. However, the most informative lineages that tell us something about how this type of photosynthesis evolved are lineages that have extant intermediate species. And these tend to be economically unimportant species, often little weedy things that no one would ever care about for any reason other than studying some esoteric type of photosynthesis. So working with non-model organisms takes us out into the field a lot. This is the Spitzkop River Valley in Namibia, where I've collected some of the plants that I work with. And here is a nice photo of one of the species that I'm working with. Very, very spiny. Another trait of model organisms is that they're easy to grow and handle, and as you can probably see from all these spikes all over this, this is not easy to grow and handle. And it's also never really been studied before, so there's absolutely nothing in the way of genomic resources. So the pros of working with non-model organisms are that it's fertile ground for discoveries, and you can get a lot of really great travel in. The cons are that you don't have any of these resources that I just spent the last couple of slides describing. So before we shift gears into exactly what I've been working on, I want to give a very, very quick primer on genomics for everybody who doesn't have a biology background. So the genome is comprised of DNA that's arranged into chromosomes. Some of it codes for proteins, little bits of the sequence like I showed you in that genome browser. Some of it codes for proteins, and these are what actually do stuff that keeps you alive. So proteins are the little molecular machines that carry out all the functions of the cell. Some are regulatory, and a lot of it does nothing. A gene, as I just mentioned, is a small coding region in the genome that codes for a protein. And genes are transcribed into a molecule that's similar chemically to DNA called RNA. The transcriptome is the set of all genes that are being transcribed, so all the genes that are turned on in a given tissue or under a given stimulus. And as I just alluded to the transcriptome is different between different tissues in a multicellular organism, different development stages, and the transcriptome changes in response to different environmental stimuli, for instance, heat shock. How do we sequence any of these things? So modern sequencing involves generating millions of short reads, usually on the order of about 150 base pairs. And then you try to put it all back together again. So it's kind of like taking a stack of newspapers and tearing it up into little pieces and trying to figure out what the various news stories of the day actually were. And to assemble these reads, we use a special type of graph called a De Bruijn graph. And without going too much into the nuts and bolts, it basically, in a De Bruijn graph, nodes in your graph are little bits of sequence of length k, which we call k-mers. And edges indicate that there's overlap between those k-mers indicated by reads from the sequencer. Genome sequencing, the genome's big and it costs a lot to sequence, especially for plants where the genome can be quite big. However, it's relatively easy to assemble, although repetitive regions kind of screw things up. Because the genome is so expensive to sequence, a viable alternative is often sequencing the transcriptome. So instead of isolating DNA, you isolate RNA, and you get just the genes that are actively expressed. And you also get a number of reads to each one that's proportional to how actively it's being expressed. So you can measure gene expression. If we have a reference genome, then all we do is we take these reads and map them onto the reference genome, and boom, you're done. If we don't have a reference genome, we have to do what's called de novo transcriptome assembly, which is the main subject of my talk here. And this is challenging because genes are expressed at wildly different levels, so the number of reads you get from different genes in the genome can be very, very different, which complicates detecting errors within the sequencer output. And also you carry higher organisms do this crazy thing called alternative splicing, where a single gene can be spliced into different RNA products. And as you can imagine, that creates all kinds of bubbles and forks and things within the assembler graph. And those bubbles and forks and everything that comes of this in the assembler graph is what I've been trying to deal with. So oftentimes when you do a de novo transcriptome assembly, what you get is a whole bunch of different assembled variants for each gene. And if what you really want is to know how many genes are there actually in the genome and maybe give me the one best sequence for each gene, that becomes a big computational problem because you don't know without a reference genome how many of these assemblies cluster to a single genetic locus, a single gene within the genome. So just to frame the problem, what I'm looking at is alignments of alignments of pairs of sequences. So this is what a sequence alignment looks like. These are two sequences here, and highlighted are all the spots at which they differ in the the base that's present there. So I don't know if you can see, for instance, here, one sequence has a G and the other one has an A. So DNA is a four-letter alphabet, A, T, C, G. And this is an alignment of two sequences. This would be two different genes that are related to each other but have evolved independently for some time in the genome. Contrast that with the kind of clumps of variant assemblies that we get out of a sequencer. So you can see that all of these sequences up here have large regions of similarity, and then some of them veer off and do other things at various points, and some of them just have a few little single base differences in here. And these would be what's called a chimeric assembly, where the sequencer has accidentally sort of woven between two genes. And some of these other ones are just partial fragmented assemblies. So this one kind of went into something else and then got screwed up and didn't make it the whole way to the length of the full gene. And over here, these are graph plots of these clusters. And these are actually some of the outputs of the program that I've been working on when it's worked well. And it doesn't always work that well at identifying a nice cluster of stuff that all looks like variants of the same gene. And so we have to represent this data as tensors to go into a neural network. So how do we do that? Well, here's the Python code that takes a pairwise sequence alignment and converts it into a two-dimensional array with the first set of sequences being 0 or 1 based on whether there's a match in the alignment, and the second one being a mismatch. And if both of them are 0, that indicates a gap in the alignment. And so basically the code here just reads in two sequences, converts them with NumPy into character vectors, and then everything downstream from that is all done vectorized, because this has to be done on really big data sets. And then I've added some additional operations here. So for instance, in image processing, one strategy that's often employed is applying a little bit of a blur to an image so that the network doesn't get bogged down in the very fine details. So I've basically blurred these arrays by passing a convolutional operation over them, kind of a sliding window average. And then if so tensors have to be the same dimensions for everything, but these alignments are not all the same length. And so if the alignment is too long, we interpolate it down to the right length. And if the alignment is too short, we just plunk it into the two-dimensional array that starts off as a 2D array of zeros. And so the array is padded with zeros at the end. So shifting gears a little bit, I'll just give a very quick primer on how neural networks work, and I know my time is relatively short, so I'll go over this pretty quickly. So this is a feed-forward neural network where you have a bunch of inputs, a couple of hidden layers of nodes, and a final output. And everything is connected. These are, in Keras, these are called densely connected layers because every input connects to every node in the first layer. The output of every node in the first layer connects to the input of every node in the second hidden layer, and so forth. So these are inputs, hidden layers, and an output. And what happens over time when you train these things is some of these edges get down-weighted or effectively removed altogether if they get down-weighted far enough. And you're left with this network of important connections between inputs mapped to outputs. And then an important variation on this for looking at sequence data, which I am, why is this not advancing, is in the form of a modification of this called a recurrent neural network, where basically you have the same thing, but now you have connections between the nodes in each hidden layer, such that information can be bounced around within a layer and retained. So you've given the node a little bit of memory. So a recurrent neural network alone could look at this sequence information that I have. But what I'd like to actually do is combine a convolutional approach so we can identify important features within the sequence. So a convolutional neural network is a very, very different sort of animal. And this gives me an opportunity to show a nice, cute picture of my cat here. And I do this because often the initial problems that are taught in neural network training classes, including the one by Andrew Ng on Coursera, are cat classifiers. So basically we have a series of filters, and these are what's actually learned by the training process. And so this would be post-training. In the past, people have painstakingly tried to design these filters. Now we just leave it up to the network to learn them. So we've got, for instance, here a filter that recognizes a cat's eye. And this filter gets passed over the image whenever the animation kicks in. There it goes. So the filter passes over the image, and when it finds an eye, it emits a different sort of output, a 1 instead of a 0. And it keeps trundling along and finds every instance of spaces within the image where that filter gets triggered. So there's another eye, and so on and so on and so on. It keeps passing along. And then we've got these other filters. So for instance, we've got a filter that may have learned from the training process to recognize the ears of a cat, and those would be triggered up here. We've got maybe another one that recognizes a nose, and so on. Now, how does this apply to my sequence problem? Well, Keras gives us a tool called a one-dimensional convolutional layer. So instead of passing over a 2D image, we can pass a convolutional layer over a sequence. So for instance, here's a sequence, and we've got a filter maybe that recognizes spikes up. This is what it's learned to recognize from training, so it would be triggered here and here. We've got another one that maybe recognizes spikes down, would be triggered over here. And you can have filters that are triggered by things that might be background as well. So this one is triggered anywhere that the sequence looks more like a straight line. And what these different filters output is a new sequence. And so the filters that I'm showing here would have an output something like this. They wouldn't be strictly 0 and 1. They would be a floating point number between 0 and 1. But the blue filter would be high here, where it was triggered. The orange would be high here, and the green would be high everywhere else. So at the end of the day, we have filters that have been triggered by various different regions within the image or within the sequence. But there's one feature of the traditional like a purely convolutional approach that makes this not so great for what I'm doing. And that is, usually you end with what's called a global max pooling layer, where basically you just get the maximum value of any of these filters that was output anywhere on the image. So you get nice high values that indicate all of these filters were tripped somewhere during scanning across the image, but you don't know how many times or where in the image they were tripped when you end with global max pooling. And so that's why I've opted for several convolutional layers and then feed that sequence as a new sort of enriched higher level sequence into a recurrent neural network. And in fact, I'm using a long short-term memory LSTM network, and I'm not going to go into the details of how that works. So here is one of the network architectures that I've played around with implemented in Keras. So first we define an optimizer. And we can pass some arguments into this as a script so that we can change these things on the fly to try different parameter settings. We've got two convolutional layers and a max pooling layer. Max pooling takes the dimensions of the sequence down. Some more convolutional layers followed by max pooling layers. This is the number of filters in each layer, and this is the size of the filter. And of course activation. And in the first layer you have to specify the input shape. And then it all feeds at the end into a bidirectional LSTM. And then finally into a dense layer that gives you one output from a sigmoid function. Where's my sigmoid? Right here. So the final output of this is a 0 or a 1. And basically what that means is I seem to have missed a slide in here. Something got lost. I didn't specifically discuss what the training data is for my problem domain, and I'm sorry I don't know where that slide went. So basically what I'm doing is we want to find variants of the same locus. So to do that, I did de novo transcriptome assemblies of species for which I have a reference genome, but then sort of pretended that we didn't have a reference genome. So you use the de novo assembly to, coupled with the reference genome, to find assemblies that map to the same. Should I just stop?
Do you wish to not take questions?
I have like one more slide.
Okay.
Yeah, so I'll wrap up quickly. So basically, I can classify things using the reference genome. So I can classify assemblies as variants of the same locus by whether they match to the same locus when we search them in the genome. And then for genuinely different genes within the genome, I just use the reference genome and find genes that have a match to each other. And because they're in a well-annotated genome, I know that they are legitimately different genes within the genome. So this is the most interesting part for me, and I have to go over it kind of quickly, unfortunately. But I've got excellent accuracy on the training data. I get accuracies of 98 or 99% with the training data. But there's less certainty in the predictions when I do it on real data that hasn't been manually classified using the reference genome. So you can see that here in these histograms. Blue is the result on the training data, and so with a really high degree of certainty, it bins things either into a value very close to 0 or a value very close to 1, with not too much uncertainty. But then on real-world data, the uncertainty is quite a bit higher, as you can see from all of these green bars in through here. So there's some kind of a disconnect between training data and real-world data that needs to be resolved. And so one of the ideas that I'm playing around with is actually simulating data for pre-training and then using transfer learning on real data. And those chimeric assemblies where the assembler has woven two genes together by mistake, those are going to pose a problem for downstream clustering because they are going to come up as variants to two different gene loci, and they're going to link clusters together that shouldn't be linked together. And so I've been toying around with a few ideas for getting around that. One of them is using graph clustering algorithms such as MCL, which basically just finds the strongly connected components within your graph. And another idea I've had is expanding this out from a binary to a multi-classifier so I can specifically address which of these sequences might be chimeric and then treat them differently in the downstream applications. And so with that, I'll just quickly say thank you and sorry for going a little bit over, and maybe I can take one or two questions if there are any.
You could probably take one.
Okay.
Give it up for Matt. Any questions? There's time for one.
So
Yep.
it's me? Or?
Yes.
Okay. So you said, thank you. You said that you have a high big data. You're dealing with big data. So how you're dealing with that in order to fit your memory? Do you have memory problems related to that?
Yeah, so I have hit memory problems at a few points along the way. Most of this stuff I'm doing on a big server. So the nodes on the server have 200 plus gigs of RAM. And so memory on the server is a little bit more available.
Okay. So.
But even so, I've been hitting some memory problems. So for instance, I had to switch over from pickling my data to storing it in H5 files because that doesn't have the memory overhead.
Yeah. So some thoughts to go around this problem is to use the Dask NumPy array, which is based on chunking the data. Then the data don't need to be loaded. Not the whole data is loaded in memory, but chunks of the data. So you may have.
What's the function that does that? Sorry.
It's a library called Dask.
Okay.
And there is an interface called NumPy Dask arrays.
Okay.
Which is So they are dealing also with HD 5 files and stuff like that.
So it can leave some of it on the hard drive and process some of it at a time. Is that what I'm understanding?
It facilitates the parallelism. Instead of loading the whole NumPy array in memory and processing it on one CPU, it's gonna load a chunk of your NumPy array and deal with the data in a distributed way.
Right. Yeah, no, that sounds very useful. I'll check
exactly,
that out.
yeah.
Thank you.
Thanks for your question. Once again, give it up for Matt.